Okay, so that is another way of indirect transmission. Okay, now indirect transmission is more contagious. What do I mean by contagious? What does the word contagious mean? Right. Yes. Not touch, can be spread. Okay, anyway. No, not dangerous. No, not dangerous, not by touch. Contagious just means it's easily transferable, or it spreads more rapidly, spreads more extensively. Okay, and like affects a large number of hosts at the same time. That's called contagious. So indirect contact, because you see, how many droplets do we release into the air when we sneeze and cough? There are a lot, and those droplets can be inhaled by so many other people. So that makes a disease very contagious, right? Okay, now the first line of defense. What do I mean by first line of defense? Like when there is war going on between two countries, the country that defends itself, the defensive country, will have lines of defenses, like the first line of army, that line of army that will meet the enemy first, face to face. And then after the enemy has defeated that first line of defense, it's the second line which then confronts the army, and then the third line, and then so on, right? So in the same way, alhamdulillah, Allah Ta'ala has given us as well at least 3 lines of defenses. The first line of defense is non-specific innate. What does innate mean? These are simple English words. Innate, I-double-N-A-T-E. What does this word mean? Anybody with a good vocabulary? Innate. This is not a biology term. This is simple common English word. You're close. You're close, but not quite there. Innate means inborn, like by birth. Yes, Yusra, yes. Okay, innate means by birth, okay? So by birth, we have this line of defense by birth. And non-specific, non-specific means like against one particular type of pathogen. Non-specific meaning against all sorts of pathogens, and not specific towards any one particular type, but it is against all sorts of pathogens. So that is called non-specific innate. One example of first line of defense, which is non-specific innate, is mechanical barrier, skin, hair inside the nose, eyes, tears. How do they protect us? Easy. Skin is an impermeable layer on the outermost surface of our body, and it is made up of dead cells and proteins, protein called collagen, I think. Yes, and then it's impenetrable by the pathogens. The pathogens cannot pass through it until and unless it's cut open. And if it is cut open, then the blood clot comes in. Remember the blood clot? The blood clot is formed when the skin is cut and the blood is exposed to the external environment. That's when you have the blood clot. So if it is cut, then the blood clot comes in. Nose hair, they trap all sorts of particles, irrespective of what type of particles they might be, but they are there to trap and clean the inhaled air, right? Eyes, tears, the tears inside the eyes, by the way, these tears have these tears are antibacterial. Okay, they kill any bacteria which comes into contact with the eyes, right? Antiseptic, right? So these are the examples of mechanical barrier. Chemical barrier. Now I'm doing this real concise way because these pages were not there, so I just condensed them. Chemical barrier. Examples. Now, mechanical barrier and chemical barrier both are included in the first line of defense. Both of them are non-specific, meaning for all sorts of pathogens, and they are present by birth. Okay, mucus. This is a sticky fluid lining our respiratory passages, and this is a sticky fluid, and therefore it also traps particles from the inhaled air. So cleans the inhaled air. Ciliary action, and therefore protecting the lungs especially. Okay, lungs are vital and very delicate. Ciliary action, these are hair-like projections on the cells lining the respiratory passages again. And what they do is that they, when they come into contact with a harmful or any foreign body, they are stimulated and they flick upwards. They flick upwards, pushing or the particle upwards towards the mouth, where that particle can be swallowed into the stomach, and the stomach will then eventually digest it, because the stomach, remember, has that strong hydrochloric acid which can kill the bacteria, and then there are protein enzymes which can kill the proteins if the pathogen has any proteins, and most of the pathogens do have proteins. So sticky mucus traps pathogens, and cilia flick them upwards towards mouth, out of the body through coughing, sneezing, and then if not swallowed, can also be coughed out, can also be sneezed out. So you must have noticed in case of flu, if the cough is productive, then you see sputum in cough, okay? Stomach acid is also a sort of chemical barrier, okay? So mechanical and chemical. Mechanical, skin, nose hair, tears of the eyes, chemical barrier, mucus, stomach acid, okay, and ciliary action. Now, second line of defense, again, second line of defense is also non-specific. This is also non-specific, meaning against all the pathogens. And basically, second line of defense is composed of phagocytes. What are phagocytes? Phagocytes are those cells which can carry out phagocytosis. Remember, I discussed phagocytosis twice before. Yes, macrophages are one type of phagocytes, but we have other types of phagocytes as well, but I will not mention them here because you'll get confused and it is not included in your IGCSE. But we have other types as well, but macrophages are one type of phagocytes that you will study, right? Phagocytes are any cells that can carry out phagocytosis. Macrophage is one of them, and yes, macrophage is a white blood cell. Then the third line of defense is specific. This is a specific. Specific meaning the response will depend upon the type of pathogen. Okay, the response will be different for different pathogens. That's why we call it a specific. Okay, lymphocytes constitute this third line of defense. They produce antibodies specific to antigens. Now, what are antigens? We will discuss that in a while. Now, let's talk about macrophages a little more in detail. By the way, we have done this already, but I made this diagram for you guys one more time. This is showing phagocytosis. Now, I want you to appreciate the difference in the structures of the two cells, okay? I like to call the phagocytes as the foot soldiers of the army, who actually, grab the pathogen and, like grab the pathogen by their collars and, actually fight with them, right? They fight with them, they get them inside, they actually do, the physical manual fighting with the pathogen. How? Can you see these membranes, projections? Now, initially it was this way, this shape. Like they have irregular shape, they can change their shapes accordingly. This changing of shape is one example. They're flexible. That's why they can squirm through the gaps in between the cells lining the capillaries, remember? Yes, so, and the same flexibility of the membrane helps them in phagocytosis as well. They protrude the edges of the membrane just like hands, and they enclose the pathogen. They enclose the pathogen this way. Can you see? They're enclosing the pathogen, yes. They're surrounding the pathogen this way, and then this was part of the macrophage membrane. Can you see? This is part of the macrophage's own cell membrane. But then this pinches separate from the cell membrane and turns into a separate vacuole, which has the pathogen enclosed within it. And sorry about this mistake in the diagram. I forgot to make the pathogen here. There should be pathogen here. Yes. So this is pinched off. This part of the membrane is pinched off, or it separates from the rest of the cell membrane of the macrophage, and turns into a vacuole within which the digestive enzymes from the macrophage are released. Can you see these tiny dots throughout the cell? These tiny dots are actually apparatus for the synthesis of these digestive enzymes. Okay, these are different organelles which help the macrophage carry out its function of phagocytosis, of synthesizing and then secreting these digestive enzymes, right? And then after the pathogen has been digested, the soluble products, end products, if they are useful for the macrophage, then the macrophage uses them. If they are useless for macrophage, they are released out of the macrophage, and then they are excreted out through the blood. Right? So this is phagocytosis. Now, lymphocytes, I like to think about lymphocytes as those soldiers who use bullets. Okay? They don't fight themselves, but they use bullets. Okay? Now, their bullets are different for different enemies, and their bullets are what we call antibodies, right? Let's talk about these antibodies. Now, another thing I would like to mention here before I move on with the lymphocytes is that there are two ways these macrophages come across these pathogens. Either directly themselves, because they are like patrolling in the flowing blood, in the blood circulation. They are looking for any pathogens. They're patrolling, like you have the policemen patrolling on the roads, to check if there's anything wrong. Same way macrophages patrol throughout the body to see if there's any cut, if there's any entry of pathogen. Do I need to phagocytose anything? Right. Another way it comes across pathogens is by lymphocytes. Okay, how? That we will discuss just now while I will explain you lymphocytes. Now, this is what an antibody looks like. This is what an antibody basically looks like, okay? This part stays constant, it stays unchanged, okay? This part of the molecule of antibody stays unchanged. This top part, this V-shaped part, this varies according to the antigen. The shape of this will depend upon the shape of the antigen, right? Now, how do the antibodies work? First, let's talk about the structure of lymphocyte as well a little. How do I make a lymphocyte? Lymphocytes are not irregular in shape like macrophages were. They are roughly circular, okay? And they have a big, bulky nucleus, okay? And most of the cell is occupied by the nucleus. You will see only a thin rim of cytoplasm. Only a thin rim of cytoplasm surrounding the nucleus. The reason why lymphocytes have such a big nucleus is because they have to synthesize a lot of antibodies. Their job is to make antibodies, and antibodies making information is stored within this nucleus. So remember, when you will do cells structures in details, different types of cells, you will realize that the structure depends upon the function heavily. Okay, so because their function is to make antibodies and they don't have to do any other work, like they don't phagocytose or they don't do anything else, so they don't need the cytoplasm as much. They need this nucleus more, because this nucleus possesses the information which is required to make those antibodies, and their function is to make antibodies, right? Okay, another thing. Antibodies are protein in nature. What sort of molecule is an antibody? An antibody is protein. And remember, all proteins are sensitive to temperature, meaning that if you expose them to extremely high temperatures, their structure will get distorted. They won't be able to work properly. Just like enzymes. Remember, enzymes would get distorted. The shape of their active sites would get distorted if exposed to extremely high temperatures, and at extremely low temperatures, they are not fast enough to work at all. Same is the case with antibodies. Antibodies are very much similar to enzymes because both of them are protein in nature, right? Okay, and also they are specific, just like enzymes. Remember, enzymes were also specific for specific proteins, specific enzymes. Like, for example, you have a different enzyme for the digestion of glucose, and different enzyme for the digestion of proteins, and different enzyme for the digestion of fats, and so on and so forth. Same is the case with these antibodies. Their shape, the shape of the antibodies will depend upon the shape of the antigens. Now let's talk about the antigens a little more in detail. Antigens are marker molecules, or molecules which actually are identification of the pathogens, okay? Pathogens are recognized with the help of these marker molecules. Why do we call them marker? Is because they mark them, like, sort of, you know, it is these molecules which are recognized by these antibodies, okay? Or even by the macrophage. Remember macrophage, how do the macrophages recognize these pathogens as pathogens? Because of their antigens on their surfaces. Remember, all the cells have molecules on their surfaces. Even our own cells have molecules on their surfaces. Because of those molecules, these lymphocytes know that, okay, these cells belong to the host itself. I don't have to produce antibodies against them because they are self cells. Apne hain, inko kuch nahi kehna, right? So there's a specific set of molecules, or there's a specific sequence of molecules which is different in different organisms. Like, I have a different set of marker molecules on my cells, and you guys have a different set of marker molecules on your cells. So that is how my lymphocytes will differentiate my cells from foreign cells. That's how lymphocytes will differentiate my cells from other cells. So as soon as the lymphocytes come across any sequence other than my sequence, they will start secreting antibodies, because they know, oh, this is an outsider. He shouldn't be here. This cell shouldn't be here. This is an outsider because the sequence of marker molecules is different. So it starts synthesizing antibodies. Okay? So after you have understood this, then there's another thing. Every lymphocyte's antibody shape would be different. Okay, every lymphocyte's antibody shape would be different. For example, these are 3 types of pathogens I have tried to make, right? You can appreciate the difference in their shape. Now, these are dome-shaped. These are dome-shaped, and these are triangular. These are rectangular, right? And so the antibodies, only that lymphocyte which can synthesize this shape of antibodies will get activated in response to this pathogen. What do I mean by this? Okay, let me talk a little more in detail. So the shape of antibodies is just suitable to fit the shape of antigens. What do we call this fitting thing? We call it complementary. Like, I would say that the shape of the antibody is complementary to the shape of the antigen. So shape of antibody is complementary. Shape of antibody is complementary. Complementary means fit, exactly fitting, like a puzzle. You know, like a puzzle, like a jigsaw puzzle. Shape of antibody complementary to shape of antigen. Right. Okay. Complementary, like the shape of the active site of enzyme was also complementary to the shape of the substrate molecule. Remember, if you remember the enzyme thing. Right. Now, after the antibodies have been produced, okay, according to the shape of the antigen, I hope you understood this point, okay? So A lymphocyte which can synthesize these antibodies will be the only lymphocyte which will get activated in exposure to this pathogen. That lymphocyte will not get activated if it comes into contact with this pathogen. Why? Because it cannot synthesize these antibodies. And that lymphocyte which produced antibody for this pathogen will not get activated if it got into contact with this pathogen as well, because it cannot synthesize these antibodies, right? So one type of lymphocyte can produce only one type of antibody and will get activated by only that pathogen for which it can synthesize that antibody. Otherwise, you know, there's no point in stimulating all the lymphocytes if they cannot synthesize the appropriate antibody, right? Synthesize means to make. I hope you know this by now. Okay, how do antibodies destroy the pathogens? Now, what does this do? Now, these marker molecules are not only used for recognition, pathogens also use them for harming our tissues. Pathogens also use them for harming our cells. So it's like, you know, these are like the weapons of the pathogens. These marker molecules, you can see them as the weapons of the pathogen. So what the antibodies do is that cap them up. So cap their weapons. So if the weapons get capped, they are no longer available to damage our tissues. Like you must have seen the shape of coronavirus, right? In many videos forwarded in groups. You have seen those spikes on its surface. God forbid, yes, those spikes are the ones with which that virus damages our lung cells. I mean, the cells lining our respiratory passages. So if the antibody can cap those spikes, that virus will not be able to harm us. So it would be rendered harmless. Yeah, just like, you know, if I cap this pen up, it won't be able to write anymore, right? Like this, it won't be able to write anymore. So if they cap their harmful parts so that they cannot harm us anymore, and then the story does not end there. After capping them up, because all the viruses have been capped up, all those pathogens, they gather together, aggregate together into one destroyed ball, a ball of destroyed pathogens, a ball of destroyed viruses. And so then they are presented to the macrophage. Hey, do you need it? Hey, macrophage, do you need it? Destroy them completely. And so the macrophage takes it from the you know, the destroyed pathogen with the antibodies maybe, and then it does this process of phagocytosis, releases digestive enzymes. If there is anything useful, it uses itself, otherwise get out into the blood. Right, so the two ways in which antibodies destroy our pathogens, one is to activate the phagocytes alert. This I just told you. It presents the deactivated pathogens to the macrophages. The second is that there's another way it kills the pathogens, the antibodies, after capping the pathogen. Then the whole complex, this is called Antigen-Antibody Complex. This is a complex. Okay, this is called an Antigen-Antibody Complex. This complex then triggers a series of reactions, and those in turn activate enzymes, again, which digest the pathogens within the blood, like extracellularly, like not within the macrophage, but within the blood. Right, now, lymphocytes will only get activated if they come into contact with the pathogen, not just all the time. Why? Because synthesis of antibodies is an expensive thing, meaning it requires energy. It requires a lot of energy as well as materials, because remember, antibodies are proteins in nature. You need a lot of protein to make these antibodies. So if the lymphocytes are all the time activated, there will be a lot of energy used, a lot of proteins and amino acids will be required. Remember, proteins are made up of amino acids. So that is why To conserve all that energy and materials, lymphocytes are activated only when needed, only when they come into contact with the pathogen. Okay? Like, for example, I made this diagram. These are a bunch of lymphocytes, and they have come into contact with this culprit, this pathogen. Now, this antibody produces some other, sorry, this lymphocyte produces some other antibody. The antibody is not matching this. It won't get activated. It will pass by just as if nothing happened. This lymphocyte also cannot produce antibodies. I mean, it can produce antibodies, but not the same shape as this pathogens, antigens. And these, same story with these. Only this lymphocyte can produce antibodies which will fit onto these antigens. So out of these, only this will get activated. This will recognize it. Oh, hey, I can make antibodies against it. Okay, I got to do this job. Now it will start making copies of itself. Start making copies of it. Lots and lots of this lymphocyte, not these, this lymphocyte. It will start multiplying. This is called mitosis. And you guys haven't done mitosis, so just a simple definition. It's just division of nucleus, producing identical cells, identical nuclear material. Lots and lots of identical copies of this. Lots and lots of identical copies of this. These are called clones. Clones, must have heard this name, clones. The Dolly sheep clone was very famous. I don't know if yes, mitosis is identical copying of cells, right? Yes. So a lot of copies are called clones. They are genetically identical cells or even organism. You can use this term even for the whole organism or just cells. Now, all of this requires some time, some little time, like, like 3 to 4 days or, even a week sometimes. This all takes time. What takes time? This recognition, which lymphocyte out of these will get activated, and then, the multiplication, mitosis. And then after these cells have multiplied, they will then synthesize the antibodies. And then those antibodies will, deactivate this pathogen. All this will take some time, right? During that time, this pathogen will multiply using your tissues, using the host's tissues, and therefore damaging the host's tissues. And so the host feels ill. The host feels symptoms. They are called symptoms. What do you understand by symptoms? These days we're coming across this word symptoms a lot. Symptoms of coronavirus. Symptoms is a medical term. It is used for what the patient feels. Symptoms in simple terms means what the patient feels, like cough, fever, shivering, yes, feelings of the patient. That's called symptoms. So the host will have, yes, host will start having symptoms. But eventually, once the number has increased and once the number of antibodies is enough to cap and deactivate all of these pathogens, eventually the person feels better. Yes, and so the lymphocytes, clones win the battle, or the antibodies win the battle, and the person feels better. Alhamdulillah, Allah करे सब patients जितने भी हैं कोरोना वायरस के सब इसी तरह से हो जाए. And their clones win the battle. Immune response. Now, immune response is the way lymphocytes react to pathogens by producing antibodies. So what is an immune response? Immune response is the way our lymphocytes react to pathogens by producing antibodies, okay? For example, I have made these clones of activated lymphocytes, okay? Now, a lot of them produce those bullets to kill the pathogen, right? A lot of them did that job. But then a small group of them is preserved by the body. They do not produce antibodies, but they are used as a stored information. Like, you guys will stay here. Why? Because we want to store this information. We might need these antibodies again in the future. So you guys stay here. So a group of cells is stored within the body which will store the information regarding the synthesis of those antibodies, right? So the next time that host, if gets exposed to the same pathogen, these cells are ever ready. You know, like they are sitting with their guns ready. You see the pathogen and there they'll fire. So these memory cells, they are like those ready soldiers. They are ready with the required bullets. And if the pathogen comes into contact with them, they will start making their copies immediately, and there will be an immediate response. What do I mean by immediate response? Like within a day, not a week, not 4 days, within a day, or even within half a day, or few hours. So that's called immediate response. Immediate response. And so these memory cells, so some of them remain in the body with the stored information regarding the antigen. Next time the same pathogen enters the body, they will be the ones activated and make clones. Such cells are called memory cells, and they will immediately produce antibodies without taking much time, because they don't need to do that job of recognizing and which lymphocyte will get activated and all of that. So they will not give enough time to the pathogen to multiply. And so the host will not feel any symptoms. Before the host feels any symptoms, these memory cells will destroy the pathogen. Okay, such a person is said to be immune, right? So a person is immune to the disease. Yes. Okay, now once we have understood how these lymphocytes work, let's move on to the vaccination. What is a vaccine? A vaccine is weakened or dead viruses or bacteria, any pathogen. Okay, they are weakened or they are dead, but they have the antigens to activate the lymphocytes, but they do not cause disease. They are there to activate the lymphocytes, but they have the antigens which are required to do that, meaning those marker molecules which are required to activate the lymphocytes to produce the antibodies and the memory cells, but they do not produce the disease. I mean, they do not cause disease. Okay, these antigens will stimulate or activate the lymphocytes, and therefore we will have memory cells, and so the person will get immune. So if next time the person is attacked, memory cells will be ready to shoot with their antibodies, and we can vaccinate both children and adults. Okay. Okay, yes. I will answer your questions. Okay, you want me to answer your questions first? Let me see. Okay. Let me check from the beginning. Okay, see, there are too many. What if there are too many pathogens in the blood and it gets full? Well, if you have too many pathogens, it depends upon how strong immunity do you have. If you're overloading your, because everything has a limit. So your immune system also has a limit. If the pathogens are beyond that limit, then of course, immune system will not be able to combat all the pathogens. So Miss, all body cells have same markers? Yes, all the body cells of the same person, of the same organism will have the same set of markers, meaning sequence of markers. It is the code. It's like a code, like you have phone numbers, like the numbers are the same, but they are in different sequence. So my phone number is different, your phone number is different. So same is the case. Marker molecules are the same, but their sequence is different in different people, in different organisms. So our cells, our lymphocytes recognize us by our sequence, like phone number. Will synthesizing antigens destroy the virus? Synthesizing antigens destroy the virus? What do you mean? What do you mean by this question? Synthesizing antigens destroy the virus? Make them harmless? No, synthesizing antibodies, you mean to say? Yes, synthesizing antibodies will destroy the virus. Okay, the mitosis is ident, yes, okay. Okay, different ways showing sickness. Does this mean that corona markers are different in shape, not being recognized by RBCs? Hala, is that a mistake? We're not talking about red blood cells here. We're talking about lymphocytes here, white blood cells and lymphocytes. Okay, red blood cells' job is only to transport oxygen and small amounts of carbon dioxide. Okay, we are talking about lymphocytes. Okay, so does this mean that corona markers are different in shape? Yes. The problem with coronavirus is that our body has never been exposed to this virus before, and so our body's lymphocytes, there are no lymphocytes in our body which can synthesize the antibody against it. So that's the problem. So these days, scientists all over the world are searching for a vaccine where they can get hold of those antibodies. Now, some clinics, some hospitals in the US, I heard Mayo Clinic is working with the plasma of the patients who have recovered. So patients who have recovered, meaning that their white blood cells or their lymphocytes have managed to synthesize antibodies against the virus. So they want to know its shape. They want to know what that antibody looks like. So maybe we can make a vaccine. And so many people are working on it, right? Let's pray that, you know, we succeed. Okay, so a person, it immunizes the pathogens. The lymphocytes remember the antibodies. Yes, so coronavirus can kill or defeat by just lymphocytes and phagocytes, right? So why people are panicking and are saying that there is no vaccine? No, we don't have a vaccine, and our lymphocytes and phagocytes, not everybody's can fight it. Not everybody can fight it, and you know, it is damaging the vital, the cells of a vital organ. So by the time our immune system manages to work against it, it kills the host, you know, because the person stops breathing. The problem is with the breathing, you know, and that's why patients need ventilators. Are lymphocytes memory cells? Yes, memory cells are a type of lymphocytes. So where are the antibodies stored after coming into use one time? After they have been used, they are then, you know, after they have performed their job, they are then just broken down by the macrophages once, you know, they have performed their function of damaging the pathogen, right? There is a news that if people take hot steam bath, coronavirus can be killed within a day. I have no idea about that. So where are the antibodies stored for next time? Antibodies are not stored. The memory cells which have the required information to synthesize, to make those type of antibodies, a small group of those memory cells are stored. Not the antibodies themselves, memory cells are stored. Okay, virus spreads really fast. Yeah, it depends upon the mode of transmission. So because the mode of transmission of this virus is airborne, like, you know, in the droplets, that's why. Blood type is code. That's why vaccines don't attack others' blood when transported, like A plus and B plus. WBCs, okay, before WBCs, try figuring it out. Why do people die if the body can make antibodies? Yeah, that's what I just explained to you, that body cannot make antibodies instantly. And that is why our immune system should be strong enough. We should be leading healthy lives, eating healthy things, okay, because our diet affects our immune system, like how actively our white blood cells can respond, right? Or antigen marker cells. Antigens are marker molecules on the surface of cells, okay? Lymphocytes are brain cells? No, brain cells are made up of neurons, okay? Though that type of cell is completely different. Lymphocytes are in the blood. WBCs also make antitoxins, right? WBCs, yeah, maybe, yes. Blood cells, okay, why are there two kinds of white blood cells? Miss my 70% Pepsi, what? Okay, memory cells, yes, memory cells. Okay, let's get back to vaccination, okay? Okay, so Yes, vitamins are good for the health of these lymphocytes. Okay, we need strong lymphocytes and we need their multiplication to be really fast. Okay, and we need good antibodies. So for that, we need to eat healthy diet. Okay, immunity. Two types of immunity. Okay, what sort of immunity? The vaccination, not the vaccination, but yeah, what sort of immunity can we get? Active and passive, right? Active basically is when the person's own lymphocytes are activated and the person's own memory cells are involved in the making of antibodies. So I mean, the person's own lymphocytes are activated. They are the ones synthesizing the antibodies, and then the person also gets the memory cells, meaning it has the information is stored within the nuclei of these memory cells. Remember, I'm not talking about the antibody getting stored. No, no, no. I'm talking about this group of cells, memory cells. These are type of lymphocytes. They will be stored and their nuclei will have the information how to make that antibody. Okay, and those memory cells will react immediately next time the host comes into contact with that pathogen, right? This is called active immunity. It is long lasting, can be lifelong. Why? Because of the memory cells, because memory cells stay within the blood for life. That is why people who are vaccinated during their childhood have that immunity lifelong. Okay, so there are two ways we can get this active immunity, either by the infection itself, because the lymphocytes came into contact with the actual pathogens and then they got activated, and then that is how they produced lymphocytes and that is how they got the memory cells, or by the injection of the weakened or the dead viruses or bacteria, right? The injection of live or dead pathogen, right? These are the two ways we can have long lasting or active immunity, right? Now, passive immunity. How is passive immunity different from active immunity? Prepared antibodies. Now, these antibodies are prepared outside of the body, meaning the host's own lymphocytes did not contribute towards the production of these antibodies. These antibodies are prepared outside the body and then just simply entered into the blood. So there are no memory cells and the lymphocytes did not participate and the lymphocytes are not activated, right? That's the basic difference between passive and active. The word passive meaning the host cells did not do anything to get it. Passive means doing nothing, right? So there are no memory cells. And since antibodies are proteins in nature, they are eventually digested away, just like any other protein molecule or the plasma proteins in the body. They are also eventually broken down. Therefore, this immunity is short term. It is short term because the antibodies do not stay in the blood for long. They are eventually broken down and then removed from the body. Okay, examples of passive immunity, how do we get? By injecting antibodies. Okay, by injecting antibodies. There is a picture here in your book. Here, this picture. Passive immunity, injection of antibodies, and then by the mother. The baby getting antibodies from mother's milk. And also while the baby is within the mother's body through placenta. Right? Now, why do babies are in need of this passive immunity is because their lymphocytes are not mature enough to produce antibodies of their own, especially during the first few months of life. So they require these pre-prepared antibodies from the mother. So before birth, they are getting through the blood, which enters the baby's body through the placenta from the mother, and then after birth, through the mother's milk. Right? That is why mother's milk is so much important and necessary for babies. And babies who are given other milk other than the mother's milk have more frequency of diseases because they don't have the required antibodies to fight the diseases. So they are the ones falling sick more frequently. Okay. Okay. Why do we require passive immunity? What if it is short term? what is the significance? I mean, what is the use of getting this passive immunity? One significance is in case of babies, right? Because they cannot synthesize their own, because their lymphocytes are not mature enough. Another importance is when you require an, okay, one advantage that passive immunity has over active immunity is that passive immunity is immediate, because you are injecting antibodies directly into the blood, and they will start damaging the pathogens, start fighting the pathogens immediately. Whereas active immunity requires some time for the activation of the lymphocytes, and then, you know, their multiplication, and then the memory cells, and then the antibody synthesis. This takes longer. This is immediate. Prepared antibodies injected into the blood, and they start working. So passive immunity is more immediate, but this is slower, but this is longer lasting, this is shorter. So in circumstances, in situations when you need immediate response and short-term, passive immunity is the solution. Okay? For example, in case of rabies virus, now this is a very lethal, dangerous virus because it attacks the neurons, the nervous tissue, the nervous cells. Okay? And damage to the nervous tissue is irreversible, and it causes paralysis forever, for life. So the person is in serious danger. So therefore, we do not allow this virus. We cannot wait for the active immunity. We cannot wait for the active immunity, because by that time, the virus would have damaged the host tissue, the host neurons. Therefore, we need passive immunity against rabies. Okay? We need to inject prepared antibodies into the blood directly before the rabies can do anything. Okay. Another example where passive immunity would be required is if you are traveling to an area where you know, there are pathogens, you know, there are pathogens for which you do not have immunity, and you have to go for some reason in an emergency. Then your best bet would be to get injectables of antibodies, passive immunity, you know? Okay, now, WHO has managed to eradicate only this smallpox from the world, okay, via its vaccination campaigns, but this is the only one, okay, smallpox. This was caused by virus and direct contact meaning touch, okay, physical contact. It used to leave its survivors with scars and blindness as well. And almost all the organs were involved in this smallpox, and this was fatal. Okay, more than 80% of people at risk were vaccinated. Okay, and WHO began this campaign in 1956. And in 30 years, almost 30 years, from 56 to 80, by 1980, there was complete eradication of a smallpox. So they declared this is smallpox to have been eradicated from the face of this earth. Okay, but by that time, it had caused a lot of damage and a lot of harm to people. But alhamdulillah, yes. Now, next, WHO has been trying since many years to eradicate poliomyelitis. In short, polio, this again is viral. And this causes permanent paralysis of parts of body. By the way, this also affects the nervous system. Okay, and so this is dangerous. It causes paralysis of parts of bodies, and Nigeria and Pakistan are the two countries which still have cases of poliomyelitis emerging, you know, every once in a while. So yeah, some countries are resisting. Paralysis, I'm sorry, paralysis is when the neurons stop working, and so parts of your body stop moving because they are not getting the impulses. Okay, because remember, limbs need impulses to move. Yes, neurons are the cells of the nervous system. Right. Then there's this other example of vaccination against measles. Again, measles is viral. Okay, usually children have this schedule of vaccination that they have to follow. right? All the children need to be vaccinated. Otherwise, they are not given admissions in schools. The schools ask for that vaccination card. Measles is one of those diseases against which all the children have to have vaccine. Now, viral measles is spread by airborne droplets. Okay, it causes a skin rash, fever, blindness, brain damage. Brain damage. Yeah, so that is dangerous. Okay. Now, this is a very important point I want to talk about. The vaccination protects not only those who are vaccinated, but also those that are not vaccinated, because fewer places for virus to breed. What do you mean by that? Because the virus can breed in the body of only an unvaccinated body. Okay, but this works only when 93% of children are vaccinated. Now, what do I mean by this? This is very important. Okay. Now, you guys only ask questions from me through the chat. I have enabled the chat so that you guys can communicate with me. But please do not use it for answering other people's questions. Only I will answer. Okay. And do not discuss anything in the chat window. Okay. So I'm now sharing my screen. I will answer your questions at the end, but let me just finish what I'm trying to say. I'm sharing my screen. I want to show you something. Okay, let me go at the start, okay, from the start, because I did not show you any pictures. Now, these are the antigens on the surface of pathogens. You can see the different shapes of the pathogens and the different shapes of the antigens on their surfaces. One is bacterium, the other is virus. Then again, I want you to appreciate the shape. You know, the same pathogen has two shapes of antigens. Can you see? One shape is triangular, the other is rectangular. Right? Now, therefore, two sorts of antibodies would be required to deactivate this pathogen, to deactivate this virus or whatever. Okay? So, okay, now this picture I got from your book. I want you to appreciate first infection with a particular type of bacteria. Now, this was the first time, so the body was not ready for this. It did not have the memory cells. So the lymphocyte that had the required antibody got activated. It has started proliferating, meaning it just started multiplying, formed a lot of clones, and therefore it has started producing antibodies. So can you see all of that took how many days? 3 days. It took almost 3 days. If the x-axis is days, right? So on the third day, the person has enough antibodies to combat the bacteria. But can you see that the bacteria had already peaked on the first day when the person started having symptoms? So that means the person has stayed sick for like 2 days. And then after 2 days, the antibodies did their job, right? Now, the person has made memory cells against this particular bacteria. Now, on the second exposure, the memory cells were already ready for this pathogen. They already had the required information for these antibodies. And so you can see that the antibodies shot up so high in exposure to just a few bacteria. Just a few bacteria, and the antibodies shot up high and never gave a chance to bacteria to multiply, right? And so the person never felt sick. Alhamdulillah, what an amazing immune system Allah has blessed us with. Okay, and now here I want you to appreciate this trend of measles in USA, in some part of USA, okay? In 1964, the vaccine was licensed, and when it got licensed, the number of measles cases dropped sharply, and it never rose up, because by 2004, it's equal to 0. Nobody's getting measles now. Why? Because now majority of the population has the required antibodies in their blood due to vaccination, because all the children are getting vaccinated when they're kids, so they are all possessing the antibodies against it. Now, these are the 4 main groups to which pathogens belong. These are the 4 major groups to which our pathogens belong. The pathogens that damage our tissues, our bodies. Number one, viruses, they are the most common. Number two, bacteria. Number three, protococysts, and then fungi. Now you would ask me, what are these protococysts and what are these fungi? Now, you are supposed to study them in the classification of organisms, okay, in that chapter. They're just simple single cells, and we're not going to go into their detailed structures at this point in time. But when you do that chapter, there you will be required to know their structures. Fungi, you know, they have those hyphae and spores and all of that. I don't know if you remember. Now, this is the point that I wanted to discuss. Now, look at this top, the top of the picture. The blue man stands for not immunized but still healthy. The yellow man stands for immunized and healthy, and red stands for not immunized, sick and contagious, right? Now, this is the population. Most of them are healthy, but they are not immunized. Now, can you see there are two red people in there who are sick, who are contagious, and they are not immunized. Now, they are spreading because they have this contagious disease, so they are spreading. And as they have a spread, because no one is immunized, the contagious disease spreads throughout the population. And now you can see only 5 are left healthy. Most of them have all gotten the infection. You know, like how the epidemic is spread in China, in Italy. Yeah, this is what happened. This is how it happened, right? Now, if some of the population gets immunized, if the vaccine comes in or they get immunized in, you know, by having the infection, now what happens is contagious disease spreads through some of the population, but what happens is that they act as buffers between the infected and sick and the healthy person. Meaning that if somebody is immunized, he can get the virus, but because he's immune, he will kill the virus. And so he won't be passing it on. You get me? And so if more people get sick, the more people's lymphocytes will get activated. And so more people's lymphocytes will produce antibodies against those viruses. And so eventually a time will come when most of the people will have the antibodies against that virus. And that's when the population stops getting sick. And that's when the population is said to be immunized. So the spread of the contagious disease is contained. Now, many people are hoping the same to happen with coronavirus, that we get immunity against it. And so, you know, if we get vaccinated, if we get the vaccine, then, you know, the vaccinated people will act as buffers between the infected people and those who are not infected. And so this is how the number of cases might decrease, you know, in the population. Right, okay. So is there any other point? Okay, this is another picture of phagocytosis. I hope phagocytosis is pretty well clear. This was a picture of macrophage from inside. You can see a lot of vacuoles enveloping the pathogens, okay? And you can see how the macrophage is extending its extensions there to grab more and more of pathogens and enclose them into its vacuole. Right, okay. Now I know it's already 9 o'clock, but just last point, I don't wanna, so now I hope you understand this point, that vaccination protects not only those vaccinated, but also those who are not, because fewer places for virus to breed. And also they act as buffers between the infected and the uninfected people. And so they prevent the spread of the virus. Virus can breed in the body of an unvaccinated body. Okay, this works only when 93% of children are vaccinated. But for this herd immunity to work, for this is called herd immunity, by the way. Oh, I'm sorry, I did not mention. You know, people getting immunized and therefore they're acting as buffers. This is called herd immunity. Herd, H-E-R-D, herd. Can you give me the marker? H-E-R-D, herd. Herd, if you know, this is an English word for a group of animals, right? But this is even used for humans. Herd immunity, like a group of humans have gotten this immunity. It's called herd immunity. I just hope that we get the herd immunity against coronavirus. Okay, autoimmune disease, really quick. Now, yes, herd immunity is what, yes, most of the people are looking for and they are hoping to achieve. I just wish that we can. Lymphocytes produce, and now what happens in autoimmune disease? Now, in autoimmune disease, these lymphocytes, they have some problem in recognizing the foreign cells. They cannot differentiate the foreign cells from the self cells. So maybe they got wrong with that sequence. You know, the lymphocytes get wrong with those sequence. So they cannot distinguish the outsiders from the self cells. And therefore they start producing antibodies against the cells of the body itself. So that's a problem, right? So that's where the lymphocytes go wrong. They get confused. Yes, and one example is, the most common example is type 1 diabetes. In type 1 diabetes, the antibodies attack the beta cells of pancreas. I don't know if you have done pancreas. Pancreas, remember, is that organ which releases digestive enzymes into the small intestine. But apart from that, pancreas has also a function of producing hormone, and that hormone is insulin. Now remember, insulin is the only hormone that can decrease the level of glucose in our blood. It is the only hormone which can decrease the level of glucose in our blood. Remember, there are many hormones which can increase the level of glucose in the blood, but there is only one that can decrease it. So if you miss a hormone that increases the blood of glucose, no problem. There are many others to substitute it. There are many others to replace it. But if something goes wrong with your insulin, there is no other hormone that can substitute it. So insulin is very essential and crucial. So the problem with the patients of type 1 diabetes is that the lymphocytes start producing antibodies against the beta cells of pancreas. Now, what are beta cells? Beta cells is a group of cells within the pancreas which produce insulin. Okay, so if these cells are damaged, they can no longer produce insulin. And so the level of glucose in the blood of that patient goes really high, you know, right after the meals. And so, which is dangerous for the cells. Remember, we studied that cells require a constant environment for their best working. And part of their environment is also the level of glucose within the tissue fluids. Remember, if you remember. So because the blood glucose level is so high, it is dangerous for the cells, and so cells can be damaged. Okay, therefore these patients have to take injectables of insulin. Now, why injectables? Can they take oral? Can they take insulin oral as a tablet? Why or why not? Why? Can they take insulin as a tablet? What did you say? Good job. Yes, you are right. Yes, you are right. Okay, they cannot take oral. It takes time to digest. यही तो मसला है. We don't want the insulin to be digested. Now, insulin is protein in nature. Yes, because it will be digested by the enzymes in the stomach. And yes, Aisha Nadeem, perfect. The stomach acids destroy it and it will lose its structure, and therefore it won't act on the, you know, it won't act on the cells, and it won't be able to lower the level of glucose in the blood. Right, yeah, because it is a hormone, and so it has to be directly injected into the blood. And they are showing this picture of a woman. No, Lubna Siddiqui, tablets will not affect the body immediately. It will take some time. No, the thing is that if you take it in the form of tablet, the insulin will be digested away. It won't work at all because it will lose its structure. Remember, it does its job because of its structure. If it loses its structure, it won't work anymore. It needs its structure to be able to be recognized by the cells. Okay, so that is why it can only be injected. An example of rheumatoid arthritis in which people body parts become inflamed because the body is attacking its joint lining. Yes, Hania, yes. Rheumatoid arthritis is another example of autoimmune disease, right? There are no tablets of insulin, Maruj. There are no tablets of insulin available. Okay, this is a picture in your book of a lady, a woman, trying to inject herself with the insulin. And yeah, patients have to do it themselves because they cannot be visiting the clinics every day. Okay, and they have to, I don't know how what the frequency of insulin is, but yeah, the patients are trained. They are trained by the doctors how to inject themselves. So then they get expert at it, they get good at it. It's not that hard, and they learn it, and then they do it. Right, yeah, before every meal maybe. Yeah. Where is it injected? Maybe thighs or arms. Okay, depending. Yeah, it's intramuscular, into the muscles. Intramuscular or intravenous? I think it's intramuscular. Yes, it's intramuscular, into the muscles. Yes, insulin is produced in the pancreas, in the beta cells of pancreas. Now, I wanted to do the end of chapter questions, but because this chapter got so long, I couldn't do. It's okay. We'll do the questions tomorrow. Okay, if you guys don't mind, I'll do these questions tomorrow. And I also pulled out a few questions from its CD. So if anybody has CD, you can go through those questions. They are very interesting questions, and we will do them together. Now, let me see, what questions do you guys have for me? The lymphocytes attack the insulin creatine destroyed, forcing the person. Yes. Okay. What is the AIDS my means? What is its symptoms? What is its full form? Can you explain autoimmune again? Okay. Autoimmune is when the lymphocytes recognizing ability, you know, the ability to recognize the foreign cells from self cells, or you can say the ability to differentiate between self cells and foreign cells is messed up, is gone wrong. And so the lymphocytes cannot differentiate self from foreign cells. And so they start treating the self cells as foreign, and they start producing antibodies against them. Right now, what exactly goes wrong? Autoimmune diseases are one of those group of diseases which even scientists have not been able to understand completely. But this is what happens. Like, lymphocytes start producing antibodies against the tissues themselves. Yes, rheumatoid arthritis is one of them. Type 1 diabetes is another. Yes, so it attacks the cells instead of protecting them. Yes, yes. Isn't herd immunity the thing? Yes. Yes, even the US is hoping on herd immunity, counting on herd immunity. Miss, what are buffers? Yes, that's a good question. Buffer is like, it takes the infection. I mean, it is taking in the pathogen into its body. The host is taking the pathogen into his body, but he's not passing it on. Why? Because he has those antibodies against the pathogen. So his body is fighting them off, and that person is not passing it on. So he is like acting as a buffer between the infected person from whom he got it, and then he is not passing it on to others. So he is behaving as a buffer. Buffer is anything, it's an English word again. It means anything that takes in the shock but does not pass it on. I hope you got me. Okay. Okay. Yeah, I got this picture from Wikipedia, Ajwa. How people make antibodies. I completed the whole chapter, and you were asking me how people make antibodies. Wow. Lymphocytes make antibodies. There's this whole process of synthesizing proteins. There's this whole sequence. It is a long process how the cells make proteins. You will do that in A levels. part of it you will do in A levels, and the rest of it you will do in your, inshallah, in your medical college. Okay, so not in IGCSE. Okay, so the exact process of making antibodies is not included. So don't Pakistani people get vaccinated against measles? Yes, they do. It is a part of the routine vaccination that children get all over the world. Yes, Pakistanis also do. Vaccination is for virus, right? And what is for bacteria? Yes, we have vaccination for bacteria as well. Vaccination is not only for viruses. Okay, it can be for viruses as well as bacteria. Okay, are those give continuously until he grows up? Yeah, if the kids are not vaccinated, then of course they are exposed to those diseases and they do not have the required antibodies, right? What is eradicated means finished? Yes, eradicated means like, yes, wiped out. Now it doesn't exist anymore. What is a smallpox or cowpox? These are different names of viruses. Okay, these are different names of those viruses. Virus is not exactly viruses, but the disease, the whole disease is called smallpox. The actual name of the virus is something else. It's like, I forgot. Yeah, you can Google it. I don't remember. It's something like Ebola something. Okay. What is the case with coronavirus, passive or active? Neither passive nor active. We don't have any vaccine for it. Okay, we don't have any vaccine for coronavirus. Just pray that we get it. Is the passive immunity given to old people? Well, yeah, it can be given. Yes. Which immunity is better? See, it depends upon the circumstance. I told you, active immunity has its own advantages and disadvantages. Passive immunity has its own advantages and disadvantages. We will solve a few questions. One of the questions is dealing with this. So it will depend upon the circumstance. It will depend upon the patient, right? Once we got vaccinated in childhood, then we don't get the disease in almost our whole life. And then in old age, we get the diseases. So till then, the vaccinated antibodies will be digested away, right? See, antibodies will be digested after a few days, okay? It is only the memory cells which will stay. Okay, now the same memory cell won't stay, but you know, they also must have their lifespan, but then the same memory cells are always present. Okay, the older ones die and the new clones survive, and that's how the memory cells stay within the body of that person for the whole life, right? Yes, rapid response is when you inject antibodies directly into the blood. It can also be injected by other organisms because the immune system of babies is, yes. Passive immunity antibodies injected, active, yes. Atika, yes, good job. Injection can stimulate RWBCs, defiance against the pathogens is active. Does not vaccination active the virus and it starts fighting? Yes, yes, okay. So is there any question? Is there any other question? Okay, what causes the symptoms of a disease? The virus. The virus damaging our tissues causes the symptoms. How much time does it take to make vaccine for a particular virus? Oh, that depends upon the virus. See, we haven't been able to make virus for AIDS as yet. HIV virus. We are still, all the universities, all the big scientists of the world are, you know, constantly trying, but they are unable because HIV keeps changing itself. It keeps changing its antigens. One day it's triangular, the other day it's, you know, rectangular, and it keeps changing and we don't know what to do with it. So hopefully COVID-19 is not that way, but okay. Okay then, assalamu alaikum wa rahmatullahi wa barakatuh.
Allah Hafiz.
Tomorrow we will do the questions of this chapter. Okay, same time, 8 to 9.